This is going to be a quick couple of videos on how the mass spectrometers in our lab work, principally the Q Exactives, with which we have two. There is also a Fusion Lumos, and this can do the same thing that the Q Exactive does, but it can do other stuff too, and maybe I'll cover that in another video another time. What you see in front of you is a schematic from the vendor, I think, of well, I got it from planetorbitrap.com, which is a really nice resource. And this is a schematic of the interesting parts of our mass spectrometers. What is a mass spectrometer? Well, I'd rather focus on what we use it for, to be honest, and how we use it. So for us, we almost always use our mass spectrometers in conjunction with liquid chromatography. And that system we then call LCMS, liquid chromatography mass spectrometry. And this gives us a kind of a 3D data, and I'll talk about how those data are generated and what they mean. But our main aim really is to measure what molecules are present in a sample and how much, or at least to be able to compare how much between different samples. So for example, if you have cancer cells growing in culture and you treat some of them with a drug and the others with a placebo, I guess, then you can compare at a molecular level the differences between the treatment and the untreated. So that's our aim, really. So the first step in the process is sample prep, actually, which I guess we'll maybe cover in other videos. But there's a few different types of biomolecule that we might want to analyze. And those involve different sample preparations. But ultimately, on the liquid chromatography side of things, we want to separate the molecules in time. And there are a number of reasons for doing that. But the critical part of the apparatus that we use to separate these things is called a column. And these are columns. Let me grab my pen. Right. So there's 1, 2, 3, 4, 5 columns there. And each one is simply a tube with some stuff inside it. And you can pump liquid in one end, and the liquid comes out the other end. And the idea is that the stuff inside the column is sticky, such that the molecules of interest stick to it under certain conditions. And then what you can do is you can change the composition of the liquid that's going into the column. And as you gradually change it, the stuff you're interested in comes off the column. So these columns up here we use for metabolomics, possibly lipidomics, a couple of these ones. And they have internal diameters on the scale of millimeters. And the one at the bottom here, this is the one we use for proteomics. And this is actually a packed emitter. So again, the sample comes in this end and it comes out this end. But actually, when it comes out of this end, this goes straight into the instrument and the sample is ionized at this point. The internal diameter of this one is about 75 microns, which is the width of a human hair, something like that. Depends on your hair. And this is what we typically use for proteomics. And this is then nano HPLC. So I like to think of HPLC like climbers on a wall. So I'm stood at the bottom of the wall with a fire hose and I'm trying to get the climbers off the wall. So I start with relatively low pressure on these climbers and start spraying them all. Presumably I spray them all equally so they all have a fair chance. And the weakest climbers will fall off first and the strongest climbers will fall off last. But the critical thing is they fall off one by one because they have different strengths. They're clinging onto the wall with different strengths. So that's the kind of principle behind most HPLC. And ultimately what you get then is a separation of your molecules over time. So this plot shows a typical HPLC run, how you would represent the data, and it's called a chromatogram. In this plot, we see absorbance because this was acquired using UV detector and not a mass spectrometer. But essentially, the system is the same with mass spectrometry. You're just detecting with a mass spectrometer instead of UV. So at some point over here, the sample was loaded on. And then gradually, you can see from the red trace here, or the one that I'm tracing over. There we are. This is the mobile phase composition. And so let's say that this was a C18 column. That means that it's got lots of kind of oily chains on the material in the columns, and anything that's hydrophobic will want to stick to those under aqueous conditions. So if you imagine that this gradient here represents the amount of organic solvent that you're running onto the column, then the more organic solvent you have, the more likely the hydrophobic molecules that are stuck to your column are to come off the column. We call that elution, and so the time they come off is called the elution time. It's also sometimes called the retention time. In fact, in mass spec, it's usually called the retention time, probably. But yeah, you can see that there are a few different molecules coming off of here one at a time. So this separation gives you more time to analyze individual ions on the mass spectrometer. It also it can concentrate them a little bit as well. So if you just have your sample spraying directly into the mass spec, you have some concentration of your ion, but then that's spread out over whatever time you're spraying your sample. Whereas in this case, you can concentrate a particular molecule into a few seconds. Also, separating out the molecules helps reduce the risk of masking, where a particularly abundant molecule is all you see on the mass spectrometer at that moment in time. If you can separate out other molecules that are co-eluting, or even just the fact that you're doing HPLC in the first place, means that you're separating these things so that you can see the lower abundant ions as well. And if the retention times are reproducible, then this can help us to say exactly what these molecules are. Typically with UV HPLC, you have a relatively pure input. It's certainly not going to be a biological, well, sometimes it'll be a biological matrix. In most cases, it's not. You've done a lot of purifications, and perhaps you've purified DNA and then digested it, and you want to know what the nucleotide composition of your DNA is, then this is a good technique to use with just the UV. Because you can actually separate the different bases on an HPLC column. And you can then see what the various bases are. Using standards, you can say what the retention time or the elution time should be and figure out which of your peaks are the various bases, and you can see any impurities there as well. The critical thing is that you don't really know if there's an impurity that's co-eluting with one of your bases, and that's where mass spec really has a bit of additional power, because you can see the different masses that are eluting at that time and not just the overall signal. This is a Q Exactive system, but it's made up of two parts. So this part on the left is the Q Exactive, that's the mass spectrometer. And this part on the right here, that is the HPLC. So that's the part we've just talked about. The columns live in this part, and then somehow they come through a tube into the front of here, unless it's the nano column, in which case the nano column sits right in front of the instrument there. And so we'll leave it there, and I'll see you in the next video.